Hello and welcome. This is Mark Gordon, cardiologist from Sioux Falls, South Dakota, and I would like to take a few minutes of your time to talk about the science of Protandim and how I got interested in this amazing product. Protandim, as we all know, is a NRF2 activator, and prior to my introduction to Protandim, I did not know what a NRF2 activator was. And I would bet that 99% of the physicians in the United States have no idea what a NRF2 activator is. And it's my hope that by the end of this discussion, all of you will be very knowledgeable in NRF2 activators and probably know more than 99% of the doctors in this country about NRF2 activators and the important role that they play in maintaining health and the contribution that they have to slowing of the aging process. So, with that, let's begin. As I said, Protandim is a NRF2 activator, and what NRF2 activators do is reduce oxidative stress, and we'll get into the cellular biology of how this works in just a moment, but first let's talk a bit about oxidative stress. If you go to the website pubmed.gov, you'll find that there's about 88,000 studies on oxidative stress, and there are about 23,000 studies on oxidative stress in specific disease states. Additionally, there have been at least 200 diseases that have had oxidative stress identified as their primary cause. Well, what exactly is oxidative stress? Oxidative stress is basically the formation of free radicals and the damage that those free radicals do to our cells. Through the normal process of metabolism that all of us go through day to day, the mitochondria in our cells are the powerhouses of the cells. They're what burn the foods and the fuels that we eat, and their byproducts or their waste are these toxic molecules called reactive oxygen species or free radicals. Free radicals can cause damage to all parts of the cell. Specifically, they can damage proteins, and all the enzymes in our bodies are proteins. So if the free radicals cause damage to the proteins, our metabolic systems may not work properly. Additionally, free radicals can cause damage to lipids, and in particular LDL cholesterol. When lipids are damaged by free radicals, they become oxidized, and it's oxidized LDL cholesterol that is very atherogenic, which means it leads to the adherence of the LDL cholesterol to the walls of the arteries, and subsequently plaque formation, which can ultimately lead to heart attack and stroke. And finally, free radicals can damage DNA. When they damage DNA, they can cause mutations, and they subsequently can lead to the formation of cancer and tumors. Now, all of us are under oxidative stress on a day-to-day basis just through normal metabolism, and there's nothing that we can do to prevent that. But there are some outside sources of oxidative stress that cause damage to our bodies as well. Some of these are shown in this slide, such as UV light, sunburn, ionizing radiation, nuclear power plants, etc., smoking, air pollution, inflammation. People that have chronic inflammatory conditions, such as rheumatoid arthritis, have a significantly higher level of oxidative stress than a normal person. Additionally, obesity. Obesity is associated with a much higher level of oxidative stress. So all of these things contribute to oxidative stress, and as you can see, just living on planet Earth exposes you to a great deal of oxidative stress, which subsequently causes the free radicals to form and causes the damage to our cells. All of this together contributes to the entire aging process. Well, here's an example of oxidative stress. This is an FLR. Some of you may know what an FLR is. Some of you may not. FLR is a funny-looking rabbit. This particular rabbit was born about a month after the Fukushima nuclear power plant in Japan had a meltdown, and they had an extreme release of radiation, and subsequently this poor rabbit was born without ears. So, how do we fight these free radicals and oxidative stress? Well, historically, we've used direct antioxidants, fruits and vegetables. Our mothers always told us, be sure to eat all your fruits and vegetables Well, they didn't know it at the time, but this was the source of the antioxidants that fight off all those free radicals. And these are what we call direct antioxidants. And direct antioxidants are good, but they're not very efficient mechanisms for dealing with all the free radicals that we're exposed to. In this slide, we see how free radicals are actually taken care of by direct antioxidants. On the right-hand part of the slide, you see a free radical molecule, and it has an unpaired electron in its outer shell. It's looking to fill that outer shell, and it will steal an electron from anything that it bumps into. And it's this stealing of the electrons that causes the damage to different parts of the cells. Well, when an antioxidant molecule is in the vicinity of the free radical, the antioxidant molecule donates an electron to that free radical. It thus neutralizes the free radical, so it can no longer do any damage to the cells. In the process, however, that antioxidant molecule is consumed. So thus we have a 1-to-1 free radical neutralization rate. One antioxidant molecule neutralizes one free radical molecule, and then the antioxidant is consumed. It can interact with no further free radicals. Indirect antioxidants, on the other hand, work by a different mechanism, and protandem causes the release of these indirect antioxidants. Indirect antioxidants, as shown in this slide, which you see this giant glob in the middle of the slide, and that is actually a molecule of superoxide dismutase, which is what Dr. McCord received the Elliott Cressman Medal for, and what he did his PhD thesis on. He was the discoverer of superoxide dismutase. Superoxide dismutase is a very large molecule, and it is produced by the cells in our body, and it is a very potent antioxidant. It will neutralize 1 million free radical molecules per second every second without being consumed. So in comparison to a direct antioxidant, we can see that this certainly is a much more efficient mechanism for dealing with free radicals. So people ask me, well, how does protandem actually work in the cell? And for some of you, this may be overkill, but for those of you that are scientifically inclined, I think it's important that we understand the molecular biology and the cell biology that's going on with protandem. There are receptors on the surface of the cells for some of the components of protandem. When these components attach to these receptors, they cause a protein called KEAP1 to release another protein, a transcriptional factor called NRF2. NRF2 is thus activated at this point, and thus the term NRF2 activator. When NRF2 is activated, it then can float from the cytoplasm of the cell into the nucleus, where it can attach to an antioxidant response element. What happens at that point is all of the genes downstream on that strand of DNA are turned on, and it causes increased production or upregulation of the production of all of the proteins that these genes code for. And to date, we know that there's probably about 600 particular proteins that are upregulated by this NRF2 activation. Well, after I learned the mechanism of NRF2 activators, I thought to myself, I wonder if there's any drugs out there that are NRF2 activators. And I actually came upon two. The first is an old drug called Ultipraz, which isn't really used much anymore, but it had been used in the past to treat schistosomiasis, which is a parasitic infection. But they found that it works by a mechanism of NRF2 activation as well. And in rodent models, it inhibited the formation of multiple types of cancers, including cancers of the bladder, blood, colon, kidney, liver, lung, pancreas, stomach, trachea, skin, and breast tissue. The problem in humans was there were significant side effects, and the chemopreventive effectiveness of this agent was not widely held to be that strong. Additionally, Ultipraz generates a free radical of its own called superoxide radical, which can be quite toxic. Despite this, they are still doing some studies on Ultipraz as a NRF2 activator in the treatment of cirrhosis, gallbladder disease, bipolar disorder, and Alzheimer's disease. The big revelation I had about NRF2 activators, however, came with this next drug called VID00001. I know a person who works at Abbott Pharmaceuticals, and I happened to send him an email asking him if he knew about Protandim and NRF2 activators, and he responded by sending me this link to this website that's shown on this slide, riatapharma.com. Riata is a small pharmaceutical company that has now partnered with Abbott Pharmaceuticals with this particular drug called VID00001. The drug company Riata is a very small drug company with a very specific focus. They make drugs that increase the body's own production of natural antioxidants. That is all they do. So with this drug VID00001, which is the one that they have that's farthest down the pipeline, so to speak, they have now completed phase 2 clinical studies in chronic kidney disease and type 2 diabetes. In these studies, they found 60% of the patients had an improvement in the severity of their kidney failure. They all had significant improvement in their glomerular filtration rates and improvement in the markers of renal function in the diabetics with renal failure. There are now ongoing studies looking at this drug and similar drugs by this company for the treatment of multiple sclerosis, lung disease, Lou Gehrig's disease or ALS, Parkinson's disease, and Huntington's chorea. So when I learned about this drug and learned that this pharmaceutical company existed solely for the purpose of producing drugs that increase the body's own production of antioxidants to fight different diseases, I got very excited. For a drug company to throw all of their money behind one mechanism, such as this, meant that there was a huge potential for this agent and similar agents. And Protandim is a natural agent that's already available today, and we don't have to wait for FDA approval. No doubt VID00001 will get approval from the FDA, and they're seeking that right now. And when they do get approval, I believe that they will send their pharmaceutical reps out in mass and speak to physicians about NRF2 activation, the importance of NRF2 activation on treating and preventing certain types of diseases that they show in their studies. When this does occur, it will increase the awareness of NRF2 activators, and I believe will do nothing but increase the usage of Protandim. So, as we said, Protandim is a NRF2 activator. There are currently 7 clinical studies available for review, and these can be downloaded on pubmed.gov if you just put Protandim in the search box. And there's now one review article looking at Protandim as a preventive agent for skin cancer. The agent Protandim has been shown to reduce the markers of cellular aging to that of a newborn in all persons of all ages, and the 5 components work synergistically to increase the production of superoxide dismutase, catalase, glutathione, and at least 400, and now probably more like 600 other protective enzymes. The interesting thing about Protandim is it causes upregulation of the good enzymes, or the proteins, and downregulation of the bad ones, the pro-inflammatory proteins. And the two work hand in hand, so you have increased production of the anti-inflammatory, decreased production of the pro-inflammatory, gives you a nice balance. The first study that I'd like to talk about is the famous T-bars study that we've seen in the video. This particular study was done at the University of Colorado, and we've all seen this slide that shows the reduction of T-bars in everybody at every age that takes Protandim. This has been repeated in individual patients. I've seen the results in some of my own patients that have taken this, and they fall right on the graph that is shown in this slide. The second study was also done at the University of Colorado, and this shows the synergistic action of the ingredients of Protandim. If you look at this next slide, you can see on the bottom the individual ingredients in Protandim and how much they increase the levels of these protective enzymes. But when the 5 are added together in the formulation of Protandim, there's an 18-fold or 1,800% increase in the production of these protective enzymes. So clearly it's not just the individual ingredients themselves, but the way that the five of them work together to cause their effect on the increased production of superoxide dismutase, catalase, and glutathione. The next study is a study that was done in muscular dystrophy. This was at Texas Tech and at Harvard, Mass General Hospital. And this next slide shows MRI scans of thigh muscles of mice. The pictures on the top are the thigh muscles of mice that received protandim, and those on the bottom are the thigh muscles of mice that did not receive protandim. The striking difference that you can see in this is the increased white areas in the untreated mice. These white areas are scar or fibrous tissue formation within the muscles. And in particular, this is a protein called osteopontin. This osteopontin is what is responsible for the scar and fibrous tissue within the muscles. With protandim, osteopontin was reduced by 35%. Following this, an organization called Cure Duchenne heard about this research and has now partnered with the company, and there are additional studies going on looking at treatments for Duchenne's muscular dystrophy. The next study that I'd like to mention to you is a skin cancer study from Louisiana State University, and I'll just direct your attention to that line that is underlined and in red, which states both skin cancer incidence and multiplicity were reduced in the mice on the protandim diet by 33 and 57%. There was a companion article that went along with this showing the mechanism of how protandim helps prevent the formation of these skin cancer lesions. And then finally, they wrote a review article at Louisiana State University looking at this mechanism as something that might be utilized in the future for prevention and treatment of skin cancer. This next study is the study that really captured my attention as a cardiologist. This was a study published in Circulation, which is the journal of the American Heart Association. It is the premier cardiology journal in the country and probably arguably in the world that publishes nothing but top-notch research related to the cardiovascular fields. The editors of this journal are the tops in their fields throughout the world, and they will not let anything get by their editorial board that is not rock-solid research done at a very reputable institution by reputable researchers. So for me to see a study on this branded supplement published in Circulation really threw me over the top, and I got very excited. I had never seen prior to this any study published in Circulation with a branded supplement. So this was a very big deal. This particular study was done at Virginia Commonwealth University, and it was a study looking at congestive heart failure and pulmonary hypertension. Pulmonary hypertension is a condition in which the pressures in the right side of the heart are elevated. The right side of the heart pumps blood into the lungs. And in this study, they induced pulmonary hypertension in an animal model. And the levels of pulmonary hypertension that they induced would cause congestive heart failure in anybody. What they showed in this study, however, was that even though they induced pulmonary hypertension in all these animals, those animals that were treated with protandim did not develop congestive heart failure. As I said, pulmonary hypertension inevitably leads to congestive heart failure, but the animals that were treated with protandim were protected against that. And when they looked further, they found that the reason why they were protected is because the heart muscle in the right ventricle in those animals treated with protandim did not develop the scar tissue or fibrous tissue formation between the muscle cells, and in particular this was osteopontin, once again, the same protein that we saw in the muscular dystrophy study. In this particular study, there was about a 50% reduction in the formation of osteopontin in those animals that were treated with protandim. Additionally, the heart output was maintained in the animals that took protandim, and there was a significant drop in those animals that did not receive protandim. And the capillary blood flow to to the heart muscle was maintained in the protandim group. So all of the protective effects were there with the protandim treated group. And this really opened the door for me to expand my thinking about the treatment of congestive heart failure. Currently, when we treat heart failure, we give drugs to treat the end result. We give drugs that dilate blood vessels, that slow the heart rate, that remove excess fluid from the body. These are the end result problems of the heart failure, but don't get to the root cause, which is the fibrosis or the scar tissue within the heart muscle. And so now finally with this study, we might be getting at the root cause. And based on the results of this study, Dr. McCord and I have had communications about some human studies that we would like to do, looking at using protandim in treatment of congestive heart failure in humans. Hopefully those will come to fruition in the near future once we find a funding source. I should also mention that that study was funded in part by the National Institutes of Health, and that the company had nothing to do with the design or the funding of that study. The next study, and this was the one that was the frosting on the cake for me, was a coronary artery bypass graft study done at Ohio State University, and this one just came out in January of 2011. Everybody probably knows somebody who's had bypass surgery, and one of the problems with bypass surgery is the vein grafts tend to start to fail after about 8 to 10 years. And the thing that leads that process off is something called intimal hyperplasia. That is increased thickness of the inner lining of the artery, or inner lining of the vein, that occurs in vein grafts when they are used to carry arterial blood. Veins were never designed to carry high oxygen content or high pressures that they're placed under when they're used as conduits or bypass grafts in coronary artery bypass graft surgery. So over a period of time, the stress of the increased pressures and the increased oxygen levels flowing through these veins causes damage to that inner lining. And what is initially 1 or 2 cells thick becomes tens to hundreds of cells thick as these inflammatory cells migrate into that intima. This slide on the left-hand side shows the progression from slide A to C of what happens when you expose veins to high oxygen content for 24 weeks. That thin line-marked intima, or the inner lining, becomes markedly thickened and becomes dozens of cells thick, and this is just over 24 weeks. On the right-hand side, though, you can see a vein that was treated with the same high oxygen content but also received protandim in the bath. That vein did not develop intimal hyperplasia or increased thickness of the inner lining of that vein. This study was done at what we call ex vivo. There were human veins, but they were outside the body in a bath. Obviously, this study is something that needs to be repeated in humans, and it will probably take years to get the result of such a study, but it's a very provocative approach to help protect vein grafts. Prior to this time, we tell our patients, eat well, take your medications, keep your cholesterol and your blood pressure under control, keep your diabetes under control, and we hope your bypass grafts last a long time. But now, potentially, we have something that we can give our patients and say, this may actually help keep your bypass grafts open a lot longer. So I'm very excited about this study and that last study on heart failure as a cardiologist, and there are so many other things that need to be looked at and will be looked at in the future. So, NRF2 activators, as I mentioned at the beginning, have not been around that long, and the first studies actually started in about 1998, and by the end of 2009 there had been about 2,500 studies on NRF2 activators. This is just going to increase in the future with pharmaceutical companies like Abbott and Reata marketing drugs that are aimed specifically at the NRF2 mechanism. And as I said, I think that's only going to increase awareness and increase the potential number of people who would be interested in taking protandim. So in summary, protandim is an affordable agent that's available now. It's clinically studied. The effectiveness has been proven. It's been published in well-respected peer-reviewed journals. You do not need a prescription for it. It's very well tolerated. It's an all-natural botanical product, and it's by far the most potent antioxidant when compared to any other supplements out there. There are more studies coming. There's at least 20 additional studies that are out there right now, some of which include looking at treatment for altitude sickness in our military, looking at high stress conditions in our Navy SEALs. There are studies going on metabolic syndrome, diabetes, prevention of rejection of pancreatic islet cell transplants, etc. The research will just continue. And the neat thing about this company is they don't fund the research. The individual universities and other organizations are the ones that fund the research. The coronary artery bypass graft study was funded in part by the American Heart Association. So clearly, there are very reputable organizations out there that are seeing the benefits of Protandim and the potential that this agent has in the future for treating a lot of the chronic diseases that we attribute to aging. At this point in time, Protandim cannot make any claims about treating, mitigating, preventing, or diagnosing any disease. But it is my hope and belief that in the near future, as more studies come out, the FDA likely will allow Protandim to make such claims. So I would like to thank you for your time and your attention, and please feel free to share this webcast with others, and I would welcome any questions that any people may have. Thank you very much for your time.